Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss pyrimidine nucleotide metabolism. As discussed in a previous video, DNA is copied during the S phase or synthesis phase of the cell cycle. Before the chromosomes can be copied, that is, before DNA synthesis happens, there should be enough substrates for DNA polymerase. And these substrates include purine nucleotides, which we discussed in the last video. Today we'll look at synthesis of pyrimidine nucleotides as well as its catabolism. So DNA polymerase, which is the enzyme that copies DNA, requires these four substrates. In the last video, we talked about how these two purine nucleotides are synthesized. In today's video, we shall look at how these two pyrimidine nucleotides will be synthesized, deoxy-CTP as well as deoxy-TTP. We shall not discuss details of all the reactions and the enzymes involved in pyrimidine nucleotide metabolism. You can get these details in various biochemistry textbooks as well as on the internet. We will focus mostly on enzymes and reactions that have medical relevance, especially with pathology as well as pharmacology. We'll see how pyrimidine nucleotide metabolism is associated with the genetic disease orotic aciduria, with cancer, as well as folate deficiency and how that causes anemia as well as birth defects. As far as pharmacology is concerned, chemotherapy is the most important association with pyrimidine nucleotide metabolism. As we shall see in a few minutes, the first pyrimidine nucleotide we make is orotic acid. And this pyrimidine nucleotide, just like the pyrimidine nucleotides you see in DNA and RNA, cytosine, uracil, thymine, these atoms here come from aspartic acid, glutamine, as well as carbon dioxide. So the pyrimidine ring atoms are derived from two amino acids and carbon dioxide, which, remember, as I mentioned in the previous video, sometimes can be written as bicarbonate, same thing. Like in purine nucleotide synthesis, the sugar part comes from ribose 5-phosphate, which is mostly made by the hexose monophosphate shunt. And the phosphates of the pyrimidine nucleotides come from ribose 5-phosphate as well as ATP. Now, the synthesis of pyrimidine nucleotides, it follows a different strategy from the synthesis of purine nucleotides that we talked about in the last video. If you remember, in purine nucleotide synthesis, the nucleobase ring is synthesized step by step on the activated sugar PRPP. However, as we shall see here, in pyrimidine biosynthesis, the pyrimidine ring, the first pyrimidine ring that is made is orotic acid. This ring is synthesized first. Okay, so we first make the ring from carbon dioxide, glutamine, and aspartic acid. And once the ring is made, then it is connected to the PRPP, to the sugar. So we make the ring first and then connect it to a sugar in case of pyrimidine synthesis. In purine synthesis, we take the sugar and make the ring on the sugar step by step. Okay, so in the first few reactions where we make orotic acid, the formation of orotic acid, what happens is we first take ATP, carbon dioxide, glutamine, and convert it into the intermediate carbamoyl phosphate, and this is done by the enzyme carbamoyl phosphate synthetase 2. Recall this intermediate carbamoyl phosphate was also an intermediate in the urea cycle. In that case, the carbamoyl phosphate was synthesized by carbamoyl phosphate synthetase 1. This enzyme here, carbamoyl phosphate synthetase 2, is the enzyme that regulates the synthesis of pyrimidine nucleotides. This enzyme is inhibited by the pyrimidine nucleotides UDP, UTP, while ATP as well as the sugar PRPP are activators of carbamoyl phosphate synthetase 2. Once we get carbamoyl phosphate, the carbamoyl phosphate is then converted to carbamoyl aspartate by reaction with aspartic acid, and this is catalyzed by the enzyme aspartate transcarbamylase or ATCase. The carbamoyl aspartate is then converted to dihydroorotate, and then the dihydroorotate is converted into the pyrimidine ring, orotic acid or orotate, shown here. Now, once the pyrimidine ring orotic acid is made, now it is added to the sugar PRPP to form the first pyrimidine nucleotide called orotidine 5' monophosphate or OMP. In the next step, the OMP is decarboxylated. Basically, this carboxyl group comes out as carbon dioxide, and that forms the nucleotide, the pyrimidine nucleotide UMP. These two reactions are carried out by a bifunctional enzyme called uridine 5 monophosphate synthase or UMPS. The first part of the reaction is catalyzed by orotate phosphoribosyl transferase, while the second part of the reaction is catalyzed by OMP decarboxylase, and both these enzymes are part of this UMPS, which is a bifunctional enzyme. Now, this reaction has an important medical significance, and that's in the disease called hereditary orotic aciduria. It's a very rare disease, and in this disease, there is a deficiency in either this part of the enzyme or this part of the enzyme or both of these parts of the enzyme, UMPS. So because of this deficiency, we could have accumulation of OMP and or orotic acid in these patients. As we shall see later, this UMP eventually becomes CTP and then DCTP, and this DCTP is required for DNA synthesis. So without these reactions, we would not be able to make DCTP, so there is interference with DNA synthesis. Now recall that the red blood cells are precursors in the bone marrow. They are very actively dividing. So because of this interference with DNA synthesis, they cannot divide, and that leads to the presence of very large immature red blood cells seen in the bone marrow. And this causes a type of anemia called megaloblastic anemia. Besides anemia, we also see a developmental delay due to the DNA synthesis interference in these babies. And when an enzyme is defective or absent in any genetic disease, then the substrate can accumulate. So in this case, OMP could accumulate or orotic acid could accumulate. And both of these are relatively small molecules and are water soluble. So orotic acid here, for example, will be excreted by the kidneys, and that massive excretion of orotic acid will lead to, in the urine, may lead to precipitation, and that precipitation can cause urinary obstruction. So once UMP is formed, now the UMP can be converted into UDP by an enzyme we talked about in a previous video called NMP kinase. The diphosphate is then converted to the triphosphate by the enzyme NDP kinase. So now we have UTP. Now UTP is a substrate for RNA polymerase. Now the UTP can be converted into CTP, and this is done by the enzyme CTP synthetase. And you can see the amine group here comes from the amino acid glutamine. So now we have CTP form and CTP, like UTP is a substrate for RNA polymerase that is used during RNA synthesis or transcription. Now the CTP that is formed can now be converted into CDP. This is because these enzymes, these NDP kinases, they catalyze reversible reactions. So the diphosphate can be converted to triphosphate, or the triphosphate can be converted to diphosphate. So that's how CTP can be made into CDP, or CDP can be made into CTP. And once we get CDP, then the enzyme ribonucleotide reductase will now form the deoxy form of CDP to give you deoxy-CDP. And now the NDP kinase will convert the deoxy-CDP into the triphosphate form deoxy-CTP. So now we have deoxy-CTP, and this is one of the pyrimidine nucleotides that we need for DNA synthesis by the enzyme DNA polymerase. Okay, so now we have one of the substrates for DNA synthesis, that is deoxy-CTP. So now the question is, how do we get the other substrate, deoxy-TTP? So now we will discuss these few reactions here in this direction. We're going to go this direction to see how we get this DTTP that is required for DNA synthesis. UDP is first converted to deoxy-UDP by the enzyme ribonucleotide reductase, and then the DUDP is converted to DUTP by the enzyme NDP kinase. Now in the next reaction, the deoxy-UTP is converted by this enzyme deoxy-UTPase, UTPase, into deoxy-UMP. So the pyrophosphate comes out, and that drives the reaction, because this pyrophosphate will break down to two phosphates, and this drives the reaction to the right. So in this way, deoxy-UTP is converted to deoxy-UMP. Now we might think that why are we doing this? Because energetically this doesn't make sense, taking a triphosphate and breaking it down to a monophosphate. So this looks like, in terms of energy, this is a wasteful reaction. However, there's a very important reason why this is done. You see, this U later will be converted to T. So by taking away the U nucleotide during DNA synthesis, reducing the concentration of U by changing it into T, what the cell does, it prevents the accidental incorporation of DUTP instead of DTTP by the enzyme DNA polymerase during replication. So a very smart strategy. Reduce the concentration of U compared to T. That way the DNA polymerase doesn't accidentally bring in U into the DNA instead of a T. So now we have deoxy-UMP, and in the next reaction catalyzed by thymidylate synthase, we'll see the deoxy-UMP will be converted into the T form deoxy-TMP. So let's look at this very important reaction where deoxy-UMP is converted into deoxy-TMP. Now notice the difference between U and T is simply a methyl group. Okay, so the T nucleobase simply is methylated U nucleobase. Very similar. So where does this methyl group come from? It comes from N5N10 methylene tetrahydrofolic acid, THF. So THF will be donating this methyl group to you to give you T. So when this THF donates its methyl group, it becomes oxidized to DHF, dihydrofolate. Now this is the only reaction that alters the net oxidation state of tetrahydrofolate. Now there's an important pharmacological association with this enzyme thymidylate synthase. Now there's an anti-metabolite drug. Basically these are drugs that mimic natural metabolites, but they interfere with a normal metabolic process. So there's an anti-metabolite chemotherapy drug called 5-fluorouracil or 5-FU. And this drug is converted into FDUMP. And FDUMP irreversibly inhibits this enzyme thymidylate synthase. So without this enzyme activity, we will not be able to get DTMP, which means basically we will not be able to get DTTP for DNA synthesis. So this drug is a chemotherapy drug and interferes with any cell that is rapidly dividing. So besides the fact that FU is converted to FDUMP, which looks like this substrate DUMP, 5-FU can also be converted to FUTP. So this looks very much like UTP, and that can be accidentally incorporated into RNA in place of UTP, and that can also disrupt RNA processing as well as protein synthesis. Now there's another anti-metabolite chemotherapy drug that is called capecitabine, brand names such as Xeloda. Now this drug, capecitabine, when it's orally administered into the body, it is metabolized into 5-FU. So what happens, the drug goes through various metabolic states by the liver and so on, and one of the metabolite that is formed is deoxyfluorouridine, and this deoxyfluorouridine is converted into 5-FU, and this is done by the enzyme thymidine phosphorylase. Now it seems like this enzyme is present in higher concentration in tumor cells compared to normal cells. So this gives this drug a little bit more specific interaction with tumor cells, because tumor cells have a higher concentration of this enzyme. So we see more 5-FU being formed in tumor cells compared to normal cells. So that will help reduce the side effects of drugs such as 5-FU. Because you should remember that any drug that interferes with DNA synthesis will also interfere with DNA synthesis not only in cancer cells but also in other rapidly dividing cells that we mentioned earlier, such as the cells in the bone marrow, the red blood cells, precursors that give you red blood cells, hair cells, skin cells, GI tract cells, and so on. Now, after donating its methyl group, this methylene THF becomes oxidized to DHF, and now this DHF has to be reconverted to THF for THF to be reused, and that is done by an enzyme called dihydrofolate reductase, DHFR. So DHFR will now convert DHF into THF. Then this THF will get its methyl group from serine, which is converted to glycine, and that's catalyzed by the enzyme serine hydroxymethyl transferase, and that will give you back this very important intermediate, vitamin-derived intermediate called N5N10 methylene THF that will donate its methyl group to you to give T. So there is a chemotherapy drug called methotrexate. This is a competitive inhibitor of the enzyme DHFR. So it prevents DHF from being converted into THF, which means eventually it will slow down or stop the production of the TTP that is required for the rapidly growing cancer cells. There's also another chemotherapy drug called pemetrexed, and this drug basically inhibits any enzyme that uses folic acid. So that would include thymidylate synthase as well as DHFR. So clearly this reaction of converting DUMP into DTMP is a very important reaction because of its association with several drugs that we see used in pharmacology. Now a few words about tetrahydrofolic acid. This is derived from the vitamin B9 of folic acid. Its main function in metabolism is to transfer one carbon units, such as CH3, CH2, OH, aldehyde group, but not carbon dioxide. Okay, remember carbon dioxide transfer is done by biotin. So besides nucleotide synthesis, folic acid is also used in several reactions. For example, in the synthesis of various amino acids such as glycine, serine, as well as methionine. There are anti-parasitic drugs such as pyrimidine, as well as antibacterial drugs such as the sulfonamides, and these drugs actually interfere in THF synthesis in parasites as well as bacteria. Now, folate deficiency clearly would affect any actively dividing cells that rely heavily on nucleotide synthesis for replication of the DNA. And that includes cells such as red blood cells as well as fetal cells. So it's not surprising, therefore, that folate deficiency is associated with anemia, that is not enough red blood cells, and interference with fetal cell division is going to cause birth defects. An example of a birth defect is spina bifida. In this condition, the neural tube that eventually forms the brain and the spinal cord in the fetus does not close. It remains exposed in the fetus. Folic acid, because of its importance for the division of fetal cells, it is often recommended as a supplement for women planning to have children to make sure there is sufficient folic acid for rapid cell division that is going on to prevent any birth defects. Okay, so now we have DTMP. So in the next reaction, this DTMP will be converted into the diphosphate form, DTDP, by nucleoside monophosphate kinase. And then finally, nucleoside diphosphate kinase will convert the diphosphate into the triphosphate. So now we have deoxy-TTP as well as deoxy-CTP. So the cell now has the two pyrimidine nucleotides it requires, the deoxypyrimidine nucleotides, for synthesizing, for copying the DNA in the S phase of the cell cycle. A few words about what happens if we have excess pyrimidines. We want to break them down. So a little bit about the catabolism of pyrimidine nucleotides. Now, unlike the purine ring, remember the purine ring is not broken down. We excrete it as uric acid. So unlike purine rings, pyrimidine rings are completely broken down. Okay, so we do break down pyrimidine rings into simpler chemicals such as beta-alanine and beta-amino isobutyrate, which are then converted into malonyl-CoA as well as methyl-malonyl-CoA. Now, malonyl-CoA, if you remember, is an intermediate for fatty acid synthesis, while methyl-malonyl-CoA can be converted into the TCA cycle intermediate, succinyl-CoA. Now, I want you to make a connection. This conversion of methyl-malonyl-CoA into succinyl-CoA, this is something we talked about in video 5.24 on the oxidation of odd-chain fatty acids. This reaction requires vitamin B12. So B12 is rarely used in metabolism. This is one of the reactions, if you recall, that requires vitamin B12. This is the reaction where we convert methyl-malonyl-CoA into succinyl-CoA. So once we get succinyl-CoA is a substrate for the Krebs cycle. It can help the cell generate energy, ATP. So pyrimidine nucleotides may contribute a small amount of energy to the cell. Now recall that in purine nucleotide metabolism, we talked about salvage pathways, where we simply take the nucleobase and connect it to a sugar to give you a nucleotide. Similarly, we do have salvage pathways for the pyrimidine nucleotides. So for example, U can be converted into UMP, which can then be converted into the other pyrimidine nucleotides. C can be attached to the sugar to form CMP, which can then be converted to DCTP. And this is done by various kinases as well as other enzymes. Okay, so now we have all the substrates necessary for DNA polymerase to copy the chromosomes. So in the next video, we shall see how these chromosomes are copied, DNA synthesis or replication. So once again, thank you very much for supporting this channel. Have a wonderful, wonderful day, and we'll see you soon.